I'm going to be talking about mating between the different Culex pipiens biotypes, which is part of something that I'm looking at for my PhD. So Culex pipiens was first described in 1758 and has subsequently been recognised as a global species complex, comprising these 4 species. So the females of these 4 species are morphologically indistinguishable from one another, however they can be differentiated based on their ecological, physiological and behavioural traits, and these then influence their ability to vector diseases. In the UK, we have the Culex pipiens species, and a recent review of all the indigenous mosquito species in the UK has highlighted Culex pipiens as a potentially important vector of diseases in the future. So the Culex pipiens species can then further be divided into 2 morphologically indistinguishable biotypes, Culex pipiens pipiens and Culex pipiens molestus, which again can be differentiated based on their ecological and behavioural characteristics. So the pipiens biotype is known to be anautogenous. They require a blood meal to produce their egg batches. They're ornithophilic, so they preferentially bite birds. They're eurygamous, they mate in open spaces and in swarms, and they're heterodynamic, meaning they undergo a winter diapause. Contrastingly, the molestus biotype does not require a blood meal to produce its first egg batch. They will preferentially bite mammals and humans. They can mate in confined spaces, and they can be active all year round. So traditionally, these 2 forms were thought to occupy distinct ecological niches, with the pipiens biotype predominantly being found above ground, whilst the molestus biotype was confined to underground environments. However, more recent evidence suggests that this habitat separation is far less rigid than previously thought, with the 2 biotypes plus hybrid populations being found from both above and below ground habitats. Hybridisation between these 2 forms has been reported from at least 12 European countries now, including the UK, with rates of up to approximately 26%. The existence of these natural hybrid populations has important implications when we are thinking about the risk of disease transmission, as these hybrids are thought to have intermediate preferences between the 2 forms. So any changes to host preference, the rate of autogeny in a population, vector competence, or the ability to forego winter diapause may all alter virus transmission dynamics. So, as an example, West Nile virus is maintained in a primary transmission cycle between its avian hosts and its maintenance vector Culex pipiens pipiens. However, in the presence of hybrid species with an intermediate biting preference, so they'll bite both birds and humans, they can act as bridging vectors which can cause virus spillover into the human population, which can then lead to cases of encephalitis. So understanding what is driving this hybridisation and determining the mating behaviours of the different biotypes is key to understanding this. So the major barrier that was thought to exist was obviously ecological isolation. However, with varying rates of hybridisation between sympatric populations across Europe, as well as the maintenance of all 3 forms within a single colony line for at least 80 generations, suggests that there are multiple barriers that exist beyond simple ecological isolation. So other barriers that have been suggested include behavioural or environmental. So, as mentioned earlier, the requirement for swarming behaviour in the pipiens biotype, or potentially there's the potential for there to be slight differences between the wingbeat frequencies of the 2 biotypes, which could alter their recognition of each other. Other barriers that have been suggested include intrinsic, including those mediated by the commensal Wolbachia strains and cytoplasmic incompatibilities, and no doubt there are also a number of genetic factors at play. However, the rate at which all of these contribute to regulating hybridisation between these 2 forms is currently unknown. More recent behavioural studies between the 2 biotypes have started to suggest that it's the female mosquitoes that are driving this mating success and are influencing this, with the females determining whether an interaction will result in a successful mating event. So what we wanted to look at is what is the mating success of homologous and reciprocal crosses of the 2 Culex pipiens biotypes from a mixed population, with the hypothesis that the homologous crosses would have a much higher mating success rate than the reciprocal ones. So to do this, we used the Brickwood colony line, which is a mixed colony line with the ratios of the 3 forms shown in the pie chart on the right. And larvae were taken from this colony and reared at a density of 200 larvae per litre, and were given 1 milligram of guinea pig pellets per larvae on alternate days. These larvae were reared in an environmentally controlled incubator at a temperature of 25 degrees with relative humidity at 50%, and they were subjected to a lighting regime of 16 to 8 hours light to dark, with an hour of dusk and dawn on either side. So following the commencement of pupation, pupae were collected daily and split into males and females, and then one male and one female pupating within a 24-hour period of each other were paired together and put into pots shown in B, and then these were put into the bigger cardboard pots shown in C and sealed with netting. These pots were then returned to the incubators for 10 days, so approximately 2 to 3 days to allow for eclosion, and then a week to allow for mating. After the 10 days, spermatheca were dissected out of the female mosquitoes, and they were observed to look for mating success. And a successful mating event was characterised by the presence of sperm, which can be seen moving around in F in the spermatheca. So after this, heads from both males and females were taken for DNA extraction and biotype differentiation by PCR. So results so far, we've currently tested 240 mating pairs, of which there was an overall mating success rate of 17%. However, statistical analysis has currently been limited due to low sample size numbers in some of these categories. So this is particularly prominent in the molestus, with the molestus pairing, with no individuals falling within this category. However, from the data we have collected so far, there's currently been no evidence of any differences between mating success between the homologous and reciprocal crosses. However, we have seen evidence that the female pipien biotype is significantly more likely to have a successful mating interaction than the molestus or the hybrids. However, not many conclusions can be taken from this currently until a full data set has been gathered. So, future work includes continuation of the single pair mating experiments. So although so far 320 mating pairs have been set up across 2 experiments, of which 240 of those were fully assessed, we still need a third round to increase sample sizes in some of these categories. So this is because we're using a mixed colony line, so the pairing of pupae is blind, with biotype differentiation only carried out retrospectively at the end of the experiments. And if we think back to the ratios of the biotypes within the Brookwood colony, the molestus biotype only makes up a small proportion, so it's only about 5 to 10% of the population, depending on which generation you're in. So it's not surprising that it's these categories that are lacking the numbers currently. So most studies similar to this would either use 2 pure colony lines and mix them, or they would collect saliva or take a leg for DNA extraction and PCR. However, this can be quite invasive and it's likely to affect their behaviour, which would be no good for studies such as this. So what we wanted to look at was a non-invasive method of biotyping the mosquitoes prior to these experiments. And then looking further ahead, we're hoping to relate this into the field, so looking at the mating success within field populations and relating it back to this work. For the non-invasive method of biotyping, we looked at DNA extraction from pupae exuvia. So when adult mosquitoes are closed, they leave behind the pupal skin on the surface of the water. And although this itself does not contain any cells or DNA, during the process of eclosion, trace epithelial cells or other cell-containing structures may be left behind. And in other insects, this is found to be sufficient for speciation. And so this has been ongoing in the background and is in the final stages of validation, and we're hoping that this will provide an easy and non-invasive method of biotyping these mosquitoes prior to these experiments. And this would allow us, so we're hoping to take this forward into the third and hopefully final round of these experiments, and we're hoping that this will allow for targeted mating of the pupae and adults, so that we can fill the numbers in where the sample sizes are lacking currently. So I'd just like to thank everyone in the entomology group at Pirbright who has helped with this, and my supervisor Mary Cameron at London School, and I will happily take questions.